The next topic is fertilization followed by embryogenesis. Now we completed gametogenesis, so the next in the sequence is fertilization. So to know fertilization, to understand the location where fertilization is occurring, we have to have an idea of the female reproductive system. So let me draw the female reproductive system. This is the duct system starting from the base. This is the vagina, the tube. Next, a funnel-like opening towards the sac-like structure called the cervix. Now both the vagina and the cervix constitute the birth canal. Now moving above it, the cervix opens into a sac which is like an inverted pear structure, and that's the uterus or the womb. And the uterus, from both sides, from the right and left side, there are tubes, two tubes, and these tubes are called the fallopian tube or oviduct. Now the fallopian tube is divided into three regions. The hind end, which ends like a funnel, is called infundibulum. Next, the bend of the oviduct, which is slightly bulged, is called the ampulla. And the last region, which is tube, nearly 10 to 12 centimeters long, the tube-like structure is called the isthmus. So the three parts of the fallopian tube are the funnel-like structure, which is facing the ovaries. I'll just draw the ovaries, which is attached by ligaments to the uterus. So this funnel-like structure, which is facing the ovaries, is known as the infundibulum. And look at the margin of the infundibulum, it is not smooth, it is thrown into foldings. I am highlighting it, see, foldings, finger-like processes. These finger-like processes called fimbriae. So that's the first part called the infundibulum. Then it takes a bend which is slightly bulged called the ampulla. The significance of ampulla is that this is the region where fertilization of the egg and sperm takes place. Then the ampulla leads into a long tube-like structure called the isthmus, and this isthmus is almost 10 to 12 centimeters long. Then it opens into the uterus. I told you the structure of the uterus, it's pear-shaped, inverted pear-shaped structure, and the uterus slowly tapers into the cervix, and the cervix finally opens into the tube-like structure called the vagina. Now the uterus is lined by three layers. What is in yellow? Let that be perimetrium. Then inner to the perimetrium is a muscular layer called the myometrium. And innermost layer is the endometrium. It is highly glandular. I am making it thick. This is in orange in color. I am indicating with orange color. This is the endometrium. So the three layers or the three inner lining of the uterus are from out to in. Outer, which is protective of the uterus, is called the perimetrium. Then inner to that is a myometrium, which is made up of muscles and helps in contraction at the time of childbirth. And the endometrium, this endometrium is only responsible for the changes during the menstrual cycle. It is not constant. Now it is thick. At certain stages, this variation in its thickness. It is highly glandular. So from out to in or from in to out, endometrium is the innermost layer, which is highly glandular, which shows cyclical changes during menstruation. Then outer to that is a myometrium, which is muscular, which helps in contraction at the time of childbirth, and the outermost layer, which is the perimetrium, helps in protecting the uterus. Now coming back to fertilization. Our topic is fertilization. Now it is through the vagina that the sperms get in. I am indicating in white. Sperms enter through the vagina, and the process by which the sperm is deposited from the penis into the vagina is known as copulation or coitus. Now millions of sperms get in through the vagina, cervix, and they reach the fallopian tube. This is a place the sperm remains for nearly 72 hours. 72 hours means for 3 days. Okay. Meanwhile, there is a release of eggs from the ovaries. See, the ovaries have the eggs at various stages of development, and by the process of ovulation, which we learn later, an egg is released. This egg is captured by the fimbriae. So the function of the fimbriae is to grab the egg, and this egg reaches here. To make it visible, I will make it red in color. So the egg is here. So at which part of the fallopian tube is the egg? At the ampulla. Simultaneously, the sperm is present, and the sperm's life in the ampulla 72 hours. If this happens simultaneously, release of egg and copulation occurs simultaneously, it results in fertilization. The egg and sperm fuses to form the zygote. So where does egg and sperm fuse? The egg and sperm fuses in the ampulla. And a very important question. Two processes are involved when fertilization occurs. When egg and sperm fuses to form the zygote. Two processes are induced. One, polyspermy is prevented, and two, the second meiotic division of secondary oocyte occurs to form the egg and second polar body. I told you that the ovum is formed at the adult reproductive life. This is when the sperm comes in contact with the egg. So if this is the egg, we know many layers surrounds the egg, namely the plasma membrane, the zona pellucida, and the corona radiata, which I took earlier. Now, the sperm makes its way. Number of sperms are present in the vicinity of the egg, millions of sperms. And I told you the acrosome. The acrosome or the sperm license enables the sperm to penetrate through these layers to the corona radiata, zona pellucida, and plasma membrane. Okay, and only one sperm succeeds. This sperm, it succeeds to make its way. See, it made its way through the corona radiata, zona pellucida, perivitelline space, and plasma membrane. Finally, it enters the egg. So this is only one successful sperm. So there are many sperms in the vicinity. Now, when this sperm head, see, look, when the sperm head just comes in contact with the zona pellucida, the very first sperm when it comes in contact with the zona pellucida, I am highlighting the contact, the sperm head. When the sperm head is in contact with the zona pellucida, signals are sent to the periphery of the egg to prevent other sperms from entering. So, polyspermy is prevented. So, when is polyspermy prevented? When the sperm head comes in contact with the zona pellucida. See, this is the plasma membrane and the pink is the zona pellucida. So, a number of sperms tries to makes its way through all the layers of all the layers surrounding the egg, and only one sperm successfully penetrates its way, and when it comes in contact with the zona pellucida, what happens is signals are sent to the periphery. It's chemical signals blocking other sperms from entering. So, that I have given in brief. Polyspermy is prevented. So, other sperms does not make its way. Only one sperm is successful to enter into the cytoplasm. Then second event that is induced due to this contact is the second meiotic division. So, just before the sperm head could come in contact with the ovum, this is called the secondary oocyte. Okay. But once the sperm head came in contact with the secondary oocyte, what happens? The second meiotic division occurs, and this becomes the ovum, and the second polar body is formed. So, the second meiotic division occurs when the sperm head comes in contact with the zona pellucida. So, once again, two events, very important, two events that occur when the sperm head comes in contact with the zona pellucida. A number of sperms are there in the vicinity of the egg, and they release the sperm license from the acrosomes to penetrate through the different layers surrounding the egg. Only one sperm successfully reaches and touches the zona pellucida. At that point, two processes are induced. One, polyspermy is prevented, that is, chemical signals are sent to the surface of the egg, preventing other sperms from entering, and second is the second meiotic division of the secondary oocyte takes place. Initially it was secondary oocyte. When the sperm comes in contact, it gets converted, that is, the second meiotic division takes place, and we get the ovum and the second polar body. And this occurs in the ampulla, the region where this fertilization occurs, ampulla. Now I am going to show so this is all about fertilization. Next we are going to see embryogenesis, that is how the zygote is going to be transformed into a full-fledged blastocyst. So we are going to see the different stages of the zygote. So as I said, ovulation took place, the egg is at this region, and what is this region called? The ampulla. Meanwhile, it is compulsory that both egg and sperm should be released simultaneously for fertilization to take place. Now, the egg and sperm meets in the ampulla. This is infundibulum. Next region is called the ampulla, the bent region. Now, what happens when the egg and sperm fuses? We know the product is zygote. So, I am zooming through this. Millions of sperms are in the vicinity, but only one sperm and egg fuses. So, successful, one sperm and egg fuses to form the zygote. So, this is the zygote. It is a single cell structure. The sperm nucleus and egg nucleus fuse. Okay? This is the zygote. Next, what happens is simple mitotic division occurs. So, we know, like arithmetic progression, what happens? The one celled becomes two celled, two celled becomes four celled, four celled becomes 8, 8 becomes 16, 16 becomes 32, 32 becomes 64. So, we are going to see these various stages, and this happens when this zygote moves from the ampulla towards the uterus. This process is occurring. So, the stages: 2 cell stage, 4 cell stage, 8 cell stage, 16 cell stage, and then we have the 32 cell stage. So, the different stages: the 2 cell stage, 4 cell stage, I will space it here, 8 cell stage, 16 cell stage, then we are having the 32 cell stage, and then we are having final stage is the 64 cell stage. So as the zygote moves through the isthmus, what is happening? Mitotic division is taking place. This mitosis is known as cleavage, and two daughter cells are produced first to form the 2 cell stage. Then each of the 2 divides by mitosis to form the 4 cell stage, then 8 cell stage, 16, 32 and 64 where it is implanted. Now the daughter cells that are produced as a result of cleavage is known as blastomeres. Now there is a name for these stages. Up till here it is called morula, or the 8th and the 16th cell stage is known as the morula. Then the 32 and the 64 cell stage is called the blastula. And to identify the 2 blastula, the 32 cell stage is known as the early blastula and 64 cell stage is called the late blastula. So once again a brief outline of embryogenesis. We saw the egg and the sperm fuses at the ampulla and to form the zygote. The zygote immediately undergoes consecutive mitotic divisions to form a 2 cell stage, 4 cell stage, 8 cell stage, 16, 32 and 64. The first stage, that is single cell, is known as zygote. The 8th and 16th cell stage is referred to as morula. The 32 and 64th cell stage are known as the blastula, early blastula and late blastula. All this happens as this structure moves through the isthmus. And this mitotic division is known as cleavage. Now finally at the 64th cell stage, it gets implanted. So to understand implantation, we have to learn the structure of a blastula, the late blastula. See, the 32 cell stage is a ball of tightly packed cells. The blastomeres are tightly packed. So let this be the 32 cell stage. From the 32 cell stage, it is going to progress into the 64 cell stage. What happens is reorganization of the blastomeres. All the daughter cells are called blastomeres. The blastomeres reorganize and they form an outer layer called the trophoblast. And the remaining blastomeres gets attached to one end to form the inner cell mass. So this is the structure of a 64 cell stage, and this gap is filled with a fluid. And this gap filled fluid structure is known as the blastocele. So once again the 3 important structures of the 64th cell stage. The blastomeres gets reorganized, they move towards the periphery and form the trophoblast. Then the remaining blastomeres hang at the animal pole end to form the inner cell mass, and the gap is filled with a fluid, and this fluid filled space is known as the blastocele. Now it is the inner cell mass that becomes the fetus or the embryo, and the trophoblast is involved in nourishment, or it is involved in placenta formation. So this is the structure I am going to place here in the uterus. It is getting implanted. So the 64th cell stage gets implanted. So it is the trophoblast, see the trophoblast which is in white gets attached. See, it gets attached to the uterine endometrium. See, uterine endometrium. Meanwhile, the uterine endometrium, the cells of the uterus divide and redivide. So what I have indicated in orange color, I am continuing that these cells divide and redivide and covers the redivide and covers the late blastula, and thus it gets embedded on the walls of the uterus. This process is called implantation. So once again, the late blastula, which is a 64 cell stage, I described the structure. What happens is, first of all, the trophoblast, the outer covering, gets attached to the uterine endometrium. Then what happens is, the uterine endometrium, the cells divide, redivide and covers the entire late blastula, and thus the late blastula gets embedded. This process of embedding the late blastula on the uterine endometrium is known as implantation. Now it is tight and fixed in the uterus. Now slowly what happens is from the trophoblast, finger-like projection arises. These finger-like projections penetrate down into the uterine endometrium and gets interdigitated in the uterine endometrium to form a very important structure. As we all know, that structure is known as the placenta. So once again, the trophoblast forms finger-like projections. Finger-like projections called, and these finger-like projections are called chorionic villi. And this chorionic villi penetrates into the uterine endometrium, and the uterine endometrium together with the chorionic villi gets interdigitated, so much intertwined, locked up to form a structure called placenta. So, and we know the function of the placenta. In brief, I am giving the function of placenta. So that embryogenesis over. Placenta, very important structure is the placenta. First, it supplies. What and all does it supply? It supplies nutrients to the developing embryo, and also it supplies oxygen. Second function, it removes what? Nutrients. What is the byproduct nutrients? Waste. And what's the byproduct of oxygen intake? Carbon dioxide. And the third function of the placenta is it is endocrine. It produces hormones, namely progesterone, very important hormone called the pregnancy hormone, traces of estrogen, HCG and HPL. So the function of the placenta, I told you the placenta is a very intimate structure that bonds the uterine endometrium with the fetus or the blastula. It is formed when the chorionic villi penetrates down into uterine endometrium and gets interdigitated, and that structural and functional unit is known as placenta. The function of the placenta: one, it supplies what? Nutrients and oxygen to the embryo. Second, it removes waste and carbon dioxide from the embryo, and lastly, it's endocrine in nature. It produces hormones like progesterone, the pregnancy hormone, traces of estrogen, then HCG and HPL. So with that, I wind up this session. I hope you have understood fertilization and embryogenesis.